to lesson 3 of module 2. This is part 3 of the autotroph series, and in this video we're going to be focusing on the mechanisms of transport. So in the last video we looked at the tissues which are responsible for transporting the substances throughout the plants. In this video we're looking at the way in which they actually do that process. The first of which is transpiration. And there are some pathways that the water takes from the roots into the plant. One of these is the extracellular pathway, and this is where most water and some mineral ions pass in or between the cells of the root. And then there's the cytoplasmic pathway, where most mineral ions and some water pass through the cytoplasm of living root cells. This involves substances entering a root hair cell by crossing the cell's membrane and then passing from cell to cell through plasmodesmata. And these are strands of cytoplasm that connect one cell with the next. So here we can see the different pathways that they may take. We can see that it does happen via osmosis, and we can read that those different pathways on the right hand side there. So the way in which water moves through the xylem can be in three main types. So the one is active transport, and that's where most dissolved mineral ions are selectively taken into the roots. There is osmosis, where the high concentration of ions in the vascular tissues of plants creates a very large osmotic concentration gradient, which allows the water to enter the roots. And then we have diffusion, where some mineral ions, including potassium and phosphate, enter the roots by diffusion. Therefore, the uptake of these nutrients depends on the rate of water uptake. There is pressure within the root as a result of this osmotic gradient, and as it continually draws in water that travels up the stem. This also causes guttation, which is the loss of liquid water through specialized pores at the end of leaf veins. As I mentioned before, the word transpiration, and this is the passive movement of water through the xylem of vascular plants from the roots to the leaf. And we have learnt before that passive does mean not requiring energy. Transpiration does also include the evaporation of water from the leaves, and that's via the stomatal pores. 99% of the water absorbed by the roots is lost via transpiration, leaving that 1% to be used for metabolic processes. The process of transpiration enables the plant to absorb any water necessary for photosynthesis, but to also transport mineral salts to the leaf cells and to fruits, and it also plays an important role in the maintenance of the temperature of the plant. So here we see a diagram which shows the water uptake by the roots and being transported up the tree through the vascular system. We can see around the outside here that there are a number of factors which can influence the rate of transpiration. And then because that transpiration rate is happening at different speeds, we will see a change in the rate of water uptake. So the way in which that water moves from the roots to the leaves is proposed or was proposed in 1894 and the theory, which is the transpiration cohesion-tension theory, it was used to explain the upwards movements of water. It does explain the primary mechanism of that movement, and there are three main ways which work together to create this theory. And the first is cohesion, which is between the water molecules. And the water molecules that are evaporated from the surface of the leaf cause a connected chain pulling water molecules from the roots, and it is constantly being replaced because of this cohesion. There is adhesion between water molecules and the plant cell walls, which causes the water not to go back down, so it's a continual stream. And then we see the tension, which is also known as a differential pressure, and this is created when the water evaporates from the leaves. These three work together to create the transpiration stream, which is a continuous one-way flow of water from the roots to the leaves. Like I just mentioned, there are a number of factors which affect transpiration rates, and that is the degree of stomata opening. So the more open, the more water that can be lost. And this does mean that the rate of transpiration is higher during the day than at night. Humidity decreases the rate of transpiration, and this is because there's a lot of water in the air with high humidity. Temperature, so an increase in temperature increases the evaporation of water from the leaves. And then air currents, which produce wind, increase the rate of transpiration, which moves water away from the leaf, increasing the rate of evaporation of water. We then move on to translocation, and this is the transport of organic solutes from the leaves to other tissues in the plant. These solutes are produced during photosynthesis. So here we're talking about the movement of organic solutes through the phloem tissue. This is done through the individual sieve tubes, and it is in one direction in each individual sieve tube, but there are bundles of sieve tubes which can transport sap in both directions. So it can be upwards and downwards. We do have two different sites in this translocation theory, and one is the source. And these are the sites where sugars are produced during photosynthesis, and it mainly is the leaves in the plant. And then we have our sinks, and this is where the sugars are translocated to. And this is mainly roots, bulbs, stems, flowers, and fruits. Translocation, unlike transpiration, is an active process, so it does require the expenditure of energy, and it's due to the incompressibility. Due to the incompressibility of water, there is a buildup of water in the phloem, which creates the gradient which draws water into the sieve cells, and this creates a fluid pressure which does move water and sugars in a different direction as required. So we can see here that there is the xylem and phloem moving next to each other, and that the movement of fluid through the phloem is because of the pumping of the sugars with water in through the osmotic gradient, through the sieve tubes, and unloaded where it is required. That does conclude part 3 of lesson 3 of the autotroph series. Tune in for lesson 4, where we're going to focus on heterotrophs.